1997 to be used as an erosion control tactic. It can be found in riparian zones in hedgerows, growing in full sun to partial shade, but it has been observed to adapt quickly to new environments, so it has been observed growing in field-like conditions as well. European dewberry is a perennial shrub with biennial canes that holds rhizomes. The stems are filled with weak prickles that are hooked and sometimes are red, which is a key characteristic of European dewberry. The leaves are trifoliate and the flowers are five-parted, and the fruits appear in a few to a cluster of these round berries that are often deep blue and sometimes waxy looking. European dewberry has gone under the radar as an invader for quite some time. It is a relatively unstudied species with no literature being published yet on any eradication or control techniques, and this is mostly in part due to its misidentification because it does look very similar to other rubus species. It, so far in New York State, is posing a threat to the native biodiversity and is even forming monocultures at the Ganondagan State Historic Site. So with that, our objectives were to first evaluate the effectiveness of various control measures, second, to evaluate differences between dewberry invaded and uninvaded areas, and third, to survey for additional populations of dewberry throughout the region. My control study is being conducted at Ganondagan State Historic Site. This area has many restored grasslands and unfortunately has been under the stress of a lot of other invasive species that they have fortunately gotten under control. In 2017, an unknown rubus was observed to be growing around their Great Brook stream. This was later identified as Rubus caecius or European dewberry, and it is now, like I said before, threatening the native landscape surrounding the stream. To begin our study, we conducted heat mapping. And to do this, we walked in 13.5 meter transects surrounding the stream and took a GPS point every 15 meters. At this GPS point, we indicated the percent cover of dewberry that was there in relation to a 1 meter square area. And then we put this data into ArcGIS to be digitized. From this, we identified that there were 7 significant populations. From this data, I moved into mapping those 7 populations more in detail, taking into more of the environmental variables such as canopy cover and the density of the dewberry that was growing there. And because of size and the environmental similarity between these sites, we selected 5 of these populations to be selected further for our study. To set up this experiment, we established similarly sized plots throughout each of the sites. We established plots that both had dewberry in them and dewberry not in them to accomplish our second objective. And for our first objective, we randomly assigned the treatments of cutting twice, just herbicide using glyphosate, and combination treatments of herbicide and cutting using glyphosate and triclopyr. And then of course we left some of the sites to have a control as well. Before the treatment was applied, I collected data through identifying all the plants that were in the plots using a 1 meter squared quadrat. I permanently marked 10 of these quadrats throughout each of the plots and estimated the percent cover of each of the plants that were in the quadrat. And I also calculated the canopy cover at each of these quadrats using the spherical crown densiometer. After the treatments were applied, I monitored the progress of each of the treatments every 2 weeks by visiting these permanently marked quadrats as before and indicated the percent cover of dewberry that was there and took into account the rate of growth by measuring the tallest cane that was present within the quadrat as well. The left image shows the control, or where there was no treatment, 4 weeks after the combination of mechanical and herbicide application. And then the right image shows the same site that had the mechanical and herbicide combination treatment of glyphosate, again, 4 weeks after application. So as you can see, the percent cover of European dewberry did go down quite a bit. And I will explain further results next. So as you can see, we did see a reduction in the percent cover of dewberry in all of the treatment areas. However, we did notice that the triclopyr herbicide using the combination of the mechanical and herbicide treatment was the most effective, and the glyphosate treatment did not really show results beyond the mechanical treatment so far. For objective 2, we did notice that the species richness was higher in areas where there was a greater canopy cover, and it also showed a trend for more native species richness where there was less rubus, but these results aren't quite yet able to be statistically proven, so we will be collecting more data on this for the future. For objective 3 of surveying for new locations, out of 10 surveys that I conducted, we did find 3 new populations. As you can see, the new populations were in Bergen and even on the SUNY Brockport campus, as well as a different population at the Green Lakes State Park. What we did find in these newer populations is that there appears to be a difference in morphology between the eastern and western populations. As the arrows indicate, the top picture shows the population that was confirmed at the SUNY Brockport campus. As you can see, the fruit appears to be different. It appears to be only a single berry, while the populations at Green Lake State Park and in the Syracuse area show a significant more cluster of berries in that. So the next steps for this study are for the treatments to be reapplied in 2021 and to continue monitoring for the effectiveness. For objective 2, we will be conducting further analyses and collecting more data for that. And we'll also be evaluating the morphological and genetic differences between these rubus caecoeus populations. I would like to thank all of those who have helped us throughout the journey, and we'll be happy to answer any questions.
Great. We have a few minutes for some questions if anyone has them. There is a kind of a comment in the chat that dewberry is encroaching at a rare native species habitat in Chittenango Falls State Park. So obviously that is not fabulous. But maybe you could provide some comments on why you think triclopyr versus glyphosate. There was so much of it. I mean, that's a huge difference in reduction in cover.
Yeah, for sure. I can't say exactly why it might have worked better, but unfortunately, triclopyr can't be used in close proximity to water.
Of course.
Of course, had to use glyphosate at most of the sites instead of the triclopyr.
Okay. So when you remove the use of triclopyr, it looks like cutting twice is actually the most effective means of control. So not even using chemical herbicide if it's in close proximity to water.
Right, yeah, that's definitely worked very well. And unfortunately, that treatment could also only be applied to some of the sites just due to landscape restrictions and the sizes of the populations. So hopefully in the future, we can try that treatment more so we can prove that better.
I do want to add something to that, though, just quickly. Although based on percent cover change in this year, the mechanical twice was most effective. It's possible that the glyphosate will have lingering effects into the spring because we actually had to apply it 2 weeks later than we planned on because of the weather. So didn't really have a chance to have a big impact. So we're hoping to see that the glyphosate treatment will be more effective this summer.
Great. So you guys plan to have a follow-up monitoring then this spring and into the summer?
Yep.
And potentially a best management practice document or something? Great. Well, I look forward to that. That's great work. Great work, Lexi and Katie. This is fabulous. So if there aren't any additional questions in the chat, we'll move on to Seneca Park Zoo Society. If you want to stop sharing, Lexi, and Will can, or Dave can start sharing his screen.
All right, thank you. You can just say Dave Will, it makes it one word.
Dave Will, yeah.
Okay, let me pull up my vision. Okay.
Makes it easy to tell you what to do, right? Like Dave will do that.
I love watching people try to type that in emails about things that I will do. Yes. So
Dave will do it.
Yep, Dave will do this. Yes. So if everybody can see my presentation.
Yep, you're all set.
Yeah, we, of course, not knowing where the world was heading, was going to mount an impressive, large outreach and awareness campaign last year. My name is David Will. I am from the Seneca Park Zoo. I'm a lead naturalist for citizen science here at the zoo, and I run a lot of our community-based programming. I try to connect a lot of higher level science to as much community partners and community residents as we can. And then I run a lot of programs to get people out and about and then kind of doing these with us so that they can go on to do them themselves. So really everything we do through my department is all driving stewardship as best we can. So we kind of excel at these kind of things. So we decided we would take part in invasive species by just getting the word out there, taking people on hikes. So one of the things that our expectations and goals were to lead a multitude, a dozen or so of early detection hikes, mostly in urban areas throughout the city of Rochester, trying to get as much of the IMAP surveying as we can in areas where we may not have seen it before. So we were looking at areas where there was a bit of a dead spot or just not as much uploads. And then we also were doing macrophyte surveys in some of those same areas that did have water bodies available to do so. As well as one of the bigger parts of this was we were going to table here on grounds at the Seneca Park Zoo. As everybody comes past, we just give them as much info as we can, as well as create our own awareness days at the zoo where the whole weekend would be devoted to that specific invasive species. And then we would have liked to have taken part in some remediation events as well, which usually on our end means water chestnut, which is one of the easier ones to get as much of our members and residents out as possible. So I feel it necessary to mention we were shut down as many other places were in this survey season, and then we were not allowed to get back to work until about June of 2020. So when we did come back, we were pretty focused on limited guests, especially on site. So that did slow down some of our numbers. A lot of my programming out in the community was shut down, mostly if I had people that would come with me. So I was able to do a lot of these with some more specific volunteers as opposed to just opening it up to the community. So I was handpicking volunteer groups as well as with limited partner availability, and we weren't able to get back into the zoo until we were well into our survey season. But I don't believe that stopped us much. So we did get down our dozen early detection hikes. We did bring about 123 people with us on these hikes. We would do pretty large routes in the city. This is an example of one of the routes here from Cobb's Hill. Most of the time we have lots of information to give everybody. We have lots of invasive species information as well as identification that we do. It's mostly a mobile class as we wander through the woods. I do an IMAP training before each hike, just kind of right there with everybody as well. So, and then of course, due to COVID-19, we were handpicking volunteer groups to come with us so that we wouldn't just open it up and kind of be out of regulation, but we were at or just under our legal obligation to have people into the woods in our area. So we did have our early detection hikes. We did submit 95 submissions to IMAP. Pretty normal invasive species you'll see in the middle of Rochester, but we were able to survey some pretty large areas, which was nice, and some parts that I didn't see were surveyed before. As opposed to our macrophyte surveys, we mostly were surveying our very own trout pond over by the Seneca Park Zoo. This one was a little harder to get done with some of our restrictions that we were having. It was a lot of handing back and forth of things, so we weren't doing this as much, but we were able to do 4 surveys. We had 31 volunteers come with us to get hands-on with that. And we had 2 different water bodies surveyed that we did not find any aquatic invasives, which was nice. Well, aquatic plant. And then some of the information tables, which we weren't able to do until 2021, but we did get done through the winter months and then up into this spring here, where we had 7 days of tabling. We're able to reach, for us, a pretty low number, but we're hoping to be more in the future. We were able to reach almost 2,000 people with invasive species information. We created graphics that you can see here. These are half-done graphics, because that's the photo I had. Invasive species that we'd be highlighting. Each time we tabled, we highlighted a new invasive species and had a general invasive species table as well. So a lot of education as they come up to us. We'd hit them with the sign on the right you see here. We had people manning this and giving a lot of extra info. And then we would take local examples, especially on grounds. We have a good amount of invasives here on grounds, where we would spread them along the second board you see on the left there, where they'd go all over the board, and then we'd have some info in the middle for them as well. So some hands-on, real life examples of invasives as people were just enjoying their time at the zoo. And those are all of the updates that we had. Our next steps that we'll be having for next year, our proposal will be very much the same, but to the extent that we thought we would be able to do in 2020. So we'll be able to table, have invasive species awareness days where we can hit the lion's share of the members and peoples that come through the zoo, which will be much larger than the number I presented a moment ago. As well as I'm really hoping to open up we're getting a lot of interest in our nature hikes this year. I have more volunteers than I know what to do with this year for my pickups and my nature hikes. So we'll be using that kind of momentum for this year as well. So we're hoping to really get this information out to as many people as we can and kind of reach down south a little bit farther than we normally would be hitting if we had like animal programs or something like that. So we've also created a couple virtual trainings for our programs as well. So we have virtual invasive species and trainings. So that is the updates from the Seneca Park Zoo. And anybody has any questions, then we're welcome to ask.
Yeah, if anyone has any questions, you can either put it in the chat or you can just unmute and ask while we're transitioning to the next speaker. I will say that the Seneca Park Zoo Society did, a phenomenal amount of work. Given COVID conditions, they were awarded, I can't remember how much they were awarded, but they didn't use all their funds. So we were able to put those back into our budget as we are trying to do a rebudget of our budget line. So the funds will not go unspent, let's just say, which is good. So thank you, Dave, and all the work that you've done, and we look forward to working with you in 2021. So next up is Jake Kearney from Monroe County Soil and Water Conservation District. Thanks, Jake. If you want to put up your presentation. And again, if you have any questions, feel free to put them in the chat. I'm looking forward to seeing this project.
Can everybody see it?
Yep.
Alrighty, perfect. So again, I'm Jake Kearney with Monroe County Soil and Water, and I will be talking about the new invasive in our area, the Japanese Angelica tree, Aurelia alata, or JAT as I will refer to it throughout the presentation. So just a quick background on JAT in the US, originally native to Eastern Asia, and then It was introduced into the US as an exotic ornamental in the early 1800s, no doubt due to its large white inflorescences that we'll look at later in the presentation. And it is closely related to and very resembling to our native devil's walking stick, also in the Aralia family, Aralia spinosa. And we're going to look at similarities between those two things also later in the presentation as well. And then previous records of devil's walking stick in the US actually have been revealed to be mis-ID'd, and they actually were JAT. And that was actually the case here in Monroe County, I believe, as the original population that was identified as JAT was thought to be devil's walking stick for nearly 3 decades until very recently. And so there's not a whole lot of research on JAT, especially both backwards, native to and in the US, since it is fairly new. We know that it possesses a lot of characteristics of a successful invader, but we still don't know a lot about its biology or control tactics to fully eradicate it from where it is. So on the left here are 2 maps from USDA, so you can see the extent of its spread in the United States. And then down here, just in New York, you see it's a big problem in the lower Hudson area and New York City area. But if we look here on a screenshot I took off IMAP, so that correlates down there. However, it is starting to encroach into the Catskill region and even some in the Adirondack region. And then, of course, our 2 records we found in Monroe County are here in red. So USDA is a little outdated as far as that goes. So IMAP is much more updated as far as where it is in New York State. And then up here in the top right, you can see the little green diamonds of where it was found in Monroe County for a closer look there. So down in Mendon Ponds and then up in Durand Eastman in Irondequoit. So like I just mentioned, a lot of characteristics of successful invaders. It reproduces both sexually and asexually. And then it outcompetes native vegetation, mostly by shading them out as it forms very dense thickets. Its growth habit is very rapid, very aggressive. And then it is also allelopathic. So if you look down in these pictures, you can see just how dense these clumps are. And you can see mostly in this right picture here, not a whole lot is growing around it due to that allelopathy. And then like most invasives, it quickly populates open areas and it really likes disturbed habitat, which is particularly concerning here in Monroe County, as it is the most urbanized and populated county in the Finger Lakes PRISM project area. So, you know, that would be a concern if it starts to spread into like the city more and other areas where it's disturbed a lot. And then over 30 native bird species have been observed eating the fruit of JAT, which allows these populations to spread really quickly and establish far from the original population, wherever that was, which is why it was important that we surveyed all the parks in Monroe County. As you saw on the map on the previous slide, the 2 areas we found that were practically on opposite ends of the county. So I'll go over what exactly you want to look for when you're looking for JAT. So it's a tall, skinny tree that can grow up to 40 feet tall. If you see here, if you can see my mouse, these skinny things are actually the stem of it. So you can actually wrap both your hands around most of these stems. That's how skinny they are. And then they, as I mentioned, grow in clumps, as you can see here. And then at the top, you can also see the large white inflorescences I mentioned, and that's going to be your most, it's what you really want to look for if you're going to ID JAT is these white inflorescences. Devil's walking stick also has it though. So what you want to look for is the lack of a main central axis here in the inflorescences. That's going to be like your tell-all, your most positive ID that you can confirm that you have JAT wherever you're looking. Another ID characteristic that's used to identify JAT versus other similar looking species is that the leaf veins will run all the way to the tips of the margins. It's kind of hard to see because the picture is small, but if you look on this leaf here, you can tell that these veins are going all the way to the edges here. But there is overlap with that and devil's walking stick as well. So it's really the inflorescences and that lack of a central axis that is going to be your big confirmation, like I said. And then other characteristic prickles along the bark, leaves, and leaflets. You can see those here and then on the leaves as well. These large bud scars are also pretty characteristic of it. In the late summer and fall, it'll get these black berries that those birds will feed on. And then these large compound leaves are pretty telling of Aralia, as well as not many trees in our area have large leaves like that. The only other one you might mistake it for might be the tree of heaven. But again, if you put all these characteristics together, you'll be able to tell that you have JAT. And then like I just mentioned, so JAT versus devil's walking stick, looking at this chart, you can see they are very, very similar, both tall and skinny, both have prickles, and both have those large leaves. So again, it's that lack of a central axis that's going to tell you have JAT and not devil's walking stick. So devil's walking stick will have an identifiable central axis. If you find something that you know is Aralia, but you don't know if it's JAT versus walking stick, and you're like, well, it kind of sort of has an axis, maybe. If you really can't tell, chances are it's going to be JAT. The main central axis will be very identifiable in devil's walking stick. And then, so these pictures here of devil's walking stick are from a population I looked at in Fredonia. And so just to me, and I think it's evident in these pictures, the prickles in devil's walking stick here look much more aggressive than they do in our JAT that we looked at and found. But again, I only looked at one population, so that might not be a characteristic to go off of, but I certainly, as soon as I got there, I certainly noticed that. And even the leaves kind of have a differing look, but again, it's just one population, so you really just want to go off of that lack of a central axis in the inflorescences. And so now I'll get into its discovery in Monroe County. So in October 2019, that was when it was first confirmed to not be devil's walking stick, and then it was JAT. You can see right here is the original first population found right off Canfield Road here in Mendon Ponds Park. And then, so at that time, the current spread was unknown. It's still, you can't say that it's known now. It's still unknown. We weren't able to survey any private residences around the park due to COVID and time restraints. But as far as all the parks go, we did heavily survey them. And we'll go over on the next slide where exactly else it was found in Monroe County. And so like I mentioned, it's the most urbanized and populated county in the project area. So we did want to make sure we checked all parks and heavily surveyed these areas, since JAT thrives in these urban areas. And then the proximity of this JAT population to Rush Oak Openings Unique Management Area is of particular concern. Also, you can see how close they are here, because that is the last remaining oak savanna area in New York State. So we really don't want JAT to get in there and start messing with that ecosystem. So here again, you can see our original population was right here in Monroe County, and after surveying, we found 4 other populations also in Mendon Ponds Park, all along the same trail. So they're all within 100 meters of each other. So as far as spread, that's not terrible. It doesn't seem to have gone past here from what we saw surveying. But you can see they're all right here along this trail. And then these green dots, if you can see them, are 22 smaller stands we also found that were too small to be called a population, but far enough away that they were recorded as their own sort of infestation stand. And those are all 100 meters from their nearest population. So again, the spread seems to be contained to right in this area at Mendon Ponds. And then the only other population we found outside of Mendon Ponds Park was at Durand Eastman Park in Irondequoit. So off Kings Highway North here, and you can see it's between 2 trails here, and then the golf course is surrounding it. So a golf course is obviously getting mowed and treated a lot, so a lot of disturbance there. So after we found this population, we surveyed there 2 more times, didn't find anything, which is good because, you know, you would expect with all the disturbance here that it would be able to spread a lot more easily there. And so now I'm just going to run through all our populations real quick. So here's again our original population, which was also our largest. It was 589 meters squared. So the start of the trailhead is actually right here, I believe, but like that's how extensive and dense this stand was. You can't even tell where the trailhead is. And then you can see here's the trail on both sides of it, large trees, smaller individuals. So it was really a very dense stand. And then population 2 was our smallest at 36 meters squared. It was right behind population 1, and for eventual treatment, both of these populations required both cutting and spray treatment, as they both had small individuals and then the larger trees. And those are both at Mendon Ponds Park as our populations 3 and 4 here. These two are right across from each other. So here's population 3, and then population 4 is right down a little slope here. So they were very close along this trail, again, because the trail is disturbed and it's mowed out here along the road and gets a lot of sun there. And then so population 3 was 240 meters squared and population 4, 322 meters squared. So again, these two are also fairly large populations, and population 4 also had a lot of swallowwort with it. So it was really just invasive heaven over there, basically. And then population 5 here is our last at Mendon Ponds. There, as you can see, there are no taller trees here, just small individuals. However, it was so spread out that it was actually a 200 meter squared size population. It basically covered all of this area. So just required foliar spray for those ones at eventual treatment. And then our final population, population 6, was the only one not at Mendon, at Durand Eastman Park. And you can see here, tall trees, smaller individuals. So that also required cutting and spray treatment. And then that was 110 meters squared. Then population 6 was actually a weird one because you can see that it's in the middle of the woods here, which is not exactly what you would think when I said open, urban, or disturbed habitat. However, this spot here, when I was there, you could see it got the most sun out of anything else in that forest. So it got just enough sun that it was able to thrive in that little area. And so now I'll run through our pre-treatment sampling. So we did a census of all tall stems, sampled the herbaceous layer, estimated canopy cover, and then did sampling for debris and ground layer, along with collecting specimens as well. So our tall stem census, it's a census, so we obviously counted all the tall stems that were present in each population. And we defined a tall stem as at least 1.3 meters tall, or the height that you take diameter at breast height at. And so we obviously collected diameter at breast height, and then we looked at phenology of each tree. Was it vegetative? Was it budding, flowering, or fruiting? And that obviously depended on the season that we surveyed during. If it was late summer, it would either be flowering, maybe some were starting to fruit. If it was earlier, then they were just budding. And then we looked at within the population, was it on the edge of a road, edge of a trail? Was it more interior, to look at if more disturbed individuals on the edges, if there was a correlation between that and DBH or phenology. And then again, looking at disturbances, was the stem previously cut or chopped? And then so for herbaceous layer sampling, we use 1 by 1 meter squared quadrats, seen in the corner here. And then we did random stratified sampling, and as long as they were 3 meters apart, that was what we used for our deciding where the quadrats go. And then we would aim for 10 quadrats for each population. However, some were very small, like population 2, we were only able to get 2 quadrats in because it was really small. And I believe the other 2 others were like 6 and 9 quadrats were only able to be used. And then for those, we would collect the bearing, the distance to the quadrant from at least 2 established objects. So for example, at Durand, there was a really large tulip tree that was the only one around the population, so we would use that. There was a boulder at Mendon Ponds to use. And that's for if we do any future sampling, we know where to put the quadra to see if the JAT was eradicated from that area. And then we looked at canopy cover percentage, percent cover of JAT and all other species present in the quadra, and then we counted the actual number of JAT stems actually present in the quadra as well. And then so for us to get a better understanding of IDing JAT versus Devil's Walking Stick, because they were so similar, we reached out to various herbariums in the United States, and a fair amount actually got back to us. So University of Connecticut, New York State Museum, Buff State, I believe also. And we were able to look at samples that they had, and then we took our own of various morphology, age, size, growth stages, etc. So we could also get a better idea, especially because there's not a whole lot of information on JAT, as I mentioned. So hopefully if anyone else is researching it or becomes a problem somewhere else that it's spreading, they'll be able to really hopefully use our samples as a way to better ID this species. And then I also took samples for Devil's Walking Stick as well from that Fredonia population. And those samples are being held at the SUNY Brockport Herbarium. And then we also sent some to the New York State Museum in Albany, which was pretty cool that we were able to help them out getting more recent samples. And so now I'll go through the treatment. So tall stems, we didn't use a basal bark or anything. We just cut those down, whether by chainsaw or handsaw, depending on size. So here is the Parks Department cutting them down. This was actually our largest tree that they cut down here on the side of the road. And those were cut immediately after pre-treatment sampling to avoid any further reproduction that season. And then so you can see here before from the front at population 1, how clear it is now. More sun will be able to get in. You can see here, more sun's getting in here where it's getting blocked up here from the back. And you can actually even see the trailhead now, so people won't miss that trail anymore. Population 3, again, just you can see it's more clear. You can see through the woods better. And then again here with population 4, all the tall stems here were cut. So there's a very noticeable difference of just how much they would outshade whatever was beneath them. And then so immediately after cutting, the stumps were sprayed with a glyphosate solution to stunt any new growth, because JAT, once they're cut, they would sprout up very fast, again, because they like disturbance, like most invasives. And then within 1 to 4 days after the tall stem cutting, smaller individuals were treated with a foliar spray, which is another glyphosate solution. So here we have Mark Quinn from Parks Department spraying all the smaller stands. And then here's population 1 2 weeks after spraying. So you can see no new growth here. And then I went out just a couple weeks ago and there was still no new growth, and the native vegetation that was here was starting to come back as well. Excuse me. So it seems that the treatment has worked. As far as right now, spring might be a little too early since we don't know a lot about JAT as far as when new growth will start. But from what I could see, I would say that the treatment did seem to work as far as any new growth and stopping the spread a good bit. And then so I was also able to age the JAT trees to see how long they were actually here. And the oldest tree was actually 30 years old at Mendon Ponds Park, and that was reproductive. And then there were several that were only a couple years younger than that. And then at Durand, the oldest was 16 years old, and that was also reproductive. So both stands of these trees had been around for a while. Like I said, it was a misidentification at Mendon Ponds. Durand Eastman, we're not really sure where that one came from. But so they had been in the county for a pretty long time. And then education and outreach. So obviously due to COVID, we were not able to have a nature walk with the public through Mendon Ponds to educate them. However, we sent out over 400 postcards to landowners within excuse me, both areas at both parks, giving them important ID tips, background info on JAT, how to and who to report it to, and then what information we would need in order to see if we would go out there and see if it was JAT. And then so as far as future monitoring, like I said, I went out there last week and there was no new growth, but like I said, it could still be early. And that's why we really want to survey in the summer and fall again, as well as later in the spring at obviously both at Mendon and Durand for any new growth or any missed individuals. And then parks near Mendon and Durand as well, which should probably be surveyed again. Especially Rush Oak because of the oak savanna ecosystem, as I mentioned earlier. And then a big question that I had after all was said and done with this was about the Durand Eastman population. So like I said, it was in the middle of the woods in a really sunny area, allowing it to thrive, but obviously it was not planted there. So it's like I mentioned earlier, birds, lots of native birds feast on the JAT fruit. So it's possible that it came from that population and it was transmitted. But the fact that they were practically on opposite sides of the county makes that more unlikely. Chances are it's planted somewhere nearby, whether in someone's yard or maybe it was planted at Durand Eastman Park elsewhere and was since cut down. There is an arboretum at Durand Eastman. Maybe when they came through with the golf course, they cut it down. So that's a big question for me is where did that population come from? And since I still don't know half a year later, chances are I never will find out, but I'll have to live with that. And that is all for me. So if you have any questions, you can drop them in the chat or let me know.
This is great, actually, Jake. And we do have some questions. So when does JAT flower?
So it flowers in late summer. So about late July through August, it'll be flowering.
So start looking around late July.
Yep.
Is there evidence of root or stump sprouts after treatment?
So not after treatment, like I mentioned, I hadn't seen any. We noticed just if one was cut down, like when they mowed at Mendon Ponds, I could tell in there, you could see little sprouts coming out from ones that were chopped by maintenance or whatever. So that's why the stump spray was important, because we could see that after we treated it, it would probably start sprouting again fairly quickly.
So I know from speaking with other PRISMs that the lower Hudson and the Catskills region has a serious issue with JAT. They have it quite widespread. And yet this population, you know, the oldest population here is 30 years old. Do you think maybe we have different climate here and it's not able to gain hold here as well as it does downstate? Or do you think we're just missing populations?
I mean, it's possible. I know the climate isn't that much different, but it is obviously warmer down there than it is up here. They probably get less snow, less harsh climate. I would guess that somewhere we are missing populations. Like I said, we couldn't do private residency for obvious reasons. And with that Durand Eastman population, like I said, it's probably planted somewhere nearby. I know when I was talking to Katie about it, she said she thought she had heard someone planted one in an herbarium at or an arboretum at one of the parks, but I didn't. Like I said, I checked Durand's arboretum 3 times, didn't see it anywhere, all the other ones. So, but chances are it is planted somewhere and we just don't know where at the moment.
In terms of outreach or maybe to parks or other personnel, like would you recommend trainings or just brochures to the Parks Department and other, you know, nature preserves or what would be the next step?
So I think both of those are good options. Since the stand, obviously it's still at Mendon, it's not cut down, the smaller stems are still there with the prickles. So you could use that as like a hands-on sort of thing. And then also we have the specimens at SUNY Brockport. So those would be easy to show people. But obviously brochures also would be just as easy. We have a lot of pictures that we took because there weren't even a lot of pictures online. That's how little studied this species was. But so yeah, probably brochures would be best since we did obviously treat the populations and all the leaves and flowers are gone now.
How many years until JET produces berries?
That I am not sure of. I know, so the ones at Durant Eastman, like I said, were 14 and 16 years old, and those had berries. So probably by the time they're reproductive, I would imagine. I think the youngest, let me check my notes here. The youngest reproductive tree at Mendon was 5 years old. So I would imagine if that's already reproducing, the berries are going to be being produced at that time. So fairly quickly.
I wonder also if maybe the berries aren't as viable here, like maybe they don't find and take hold as well as they do downstate.
Right.
Just seems odd. For 30 years, we're not seeing more.
I know.
Yeah. So Bruce has indicated that a myrrh cork tree has popped up in several locations in Monroe County in a similar manner to JET, kind of as a FYI.
Gotcha. Never heard of that one.
Yes, I've heard it. Deb asks if it grows from suckers.
I believe it does. I'm pretty sure it does. I believe Katie, she might know that also. She wants to jump in, but I'm pretty sure it does. Yes.
Yeah, so another one to keep our eyes out for. Jake, this is a great presentation. The work that you guys did on this project is quite extensive. So we really appreciate all that you've done for us. And we look forward to publishing on our website the reports.
Thank you.
Documenting all of it. Great. So if you have additional questions, pop it in the chat. I think we'll have some time at the end for any missed questions. But at this point, we'll move forward towards Will in the Genesee Land Trust, and we'll look at that project.
Hi, I'm Will Macaluso, a stewardship assistant for Genesee Land Trust. Get this going here. So we did some invasive plant management and outreach at our Macyville Woods Nature Preserve, which is a new-ish for us nature preserve in Sodas Point. We acquired it in 2019. The preserve is about 34 acres. 2/3 of it is a beech oak hardwood forest, and then another 10 acres, 1/3 of it is freshwater wetland. Being in Sodas Point and right on the lake in Sodas Bay, it's important stopover habitat for migratory birds, which is part of the impetus for us to protect the habitat there. The understory of the preserve is largely dominated by invasive shrubs, multiflora rose, Japanese barberry, non-native honeysuckles, and the one that stood out to us that we really focused on for this project was Japanese wineberry, which, if you're not familiar with it, is a non-native raspberry species that was brought over from Asia in the 1800s, actually, to be bred with our native raspberry plants to sweeten them up. So a lot of like raspberries you would buy at the grocery store are a hybrid that might include this species. So through the PRISM grant, we removed a lot of the invasive shrubs and then replanted with native trees and shrubs, including 5 outreach events for the public, which we had to modify a little bit due to COVID, but we still were able to reach a good amount of people. So this is just a map of the preserve. You can see the area highlighted in light gray is our total treatment area. The darker gray is where we really focused most of our invasive shrub removal efforts. It seems that the area near the trailhead and along the trail is the most heavily invaded area. But we also wanted to try to improve the vertical structure diversity throughout the hardwood forest, so that a lot of the plantings went throughout the treatment area, even if we didn't remove a lot of invasive shrubs in those areas. So we removed honeysuckle and multiflora rose and Japanese barberry. The staff removed by cutting and painting the stumps. Wineberry was removed throughout the season. The largest effort was in August when we had a volunteer event where we felt comfortable having volunteers with masks working outside. We also, in the spring, when we weren't sure that we could have volunteers working with us, created a couple of videos that we hosted on our website to teach people what wineberry was and how to remove it. So we had some volunteers that were going out on their own time early in the pandemic and removing the wineberry for us. Overall, that dark gray area that I showed before was about 5 acres that we treated for invasive shrubs. To replace the non-native plants that we were removing, we planted about 300 native trees and shrubs. The majority of those were donated by Wayne County Soil and Water Conservation District, and an additional 80 to 100 were donated by the Rochester chapter of the Sierra Club, who came out and helped us plant them on October 31st. So that overall area that I showed before, the restoration area, is about 15 acres. For outreach, we had the volunteer video that I mentioned earlier, as well as 3 volunteer events. We hosted our Youth Environmental Ambassadors, which is a group of youth from Rochester that helps work on our preserves and some other lands that we steward. So the ambassadors were out. This is a picture of them against our tree that we call the Macy tree, a large tree on the preserve. They had a hike of the trails and learned about native versus non-native species and their importance, and then helped to remove some Japanese wineberry. We also had the Sierra Club come out with RIT's National Technical Institute for the Deaf. That's the picture that I showed earlier in October, where they helped with the tree planting. And then in August, we had a wineberry pick and pull. So we had several volunteers out, and we spent about an hour picking ripe berries off of the wineberry plants and bagging those. The volunteers could bring them home and eat them or make pie or whatever they wanted to do. And then we spent a couple hours pulling and bagging the wineberry plants to try to get them out of there. Our last event was in November. We led an invasive species awareness hike. It was a cold, snowy day in November, but we still had 8 people brave the weather and come out and had a nice hike of the trails and talked about invasive species and why we care if a species is native or non-native and the impacts and all that kind of stuff. So that's about it. These are just a few pictures from our wineberry pick and pull volunteer day. And if anybody has any questions or comments, I'd be happy to take them.
Great. This is a wonderful little preserve. Actually, my in-laws live on Seaman Street, so I've been on this preserve a bit. It's really great. But I'm not familiar with the wineberry on the trail. Can you maybe describe wineberry and
Sure, yeah. So you can kind of see it here in this picture, but it looks a lot like raspberry, our wild black raspberry, but it has kind of a red fuzzy stem. That's pretty indicative of it, wineberry. I don't see it in many places, which is why we thought it was kind of important to focus on it in this preserve. I've seen it here, and I've seen it at our Cornwall Preserve in Pulteneyville. It's just a little bit west of this preserve. So it's pretty, when you see the red fuzzy stems, I should have included a picture of that, but it's pretty indicative of the wineberry plant.
So the wineberry isn't very prevalent. You don't see it in too many locations.
I have not. Before I moved back here to New York, I was working in Southeast Pennsylvania, and wineberry and spotted lanternfly were both everywhere. So everybody down there knows about it. I'm assuming that it's probably moving north, but it's kind of funny that it seems like it's landed on the lake shore. So.
Yeah, Bruce has indicated that it's common at Teganic Falls State Park, so maybe it's just underreported. I just wonder, there's a lot of bramble throughout our trail systems, and for our trail survey, would we want people to dig deeper to see if those are, wild raspberry, or could it be wineberry or dewberry? That's certainly something that, not understanding the prevalence of either of those two species it's possible we can get our volunteers to try to key out.
Yeah, and I think it's a pretty easy plant to identify for a volunteer, with the stems and the berries come at a different time than our native raspberries. And they kind of start out orange where they're pretty noticeable and then mature to red. So that's another good kind of ID method.
Can you, during the next presentation or before this is up, do you have that video that you can pop in the chat? Maybe we can.
It's not on our website anymore. We removed it, but I can see if I can find it and share that.
Yeah, that might be interesting. Maybe we can highlight it on social media when it's blue, when it would be in flower or in fruit.
Right. I think ideally it's best to remove it before the fruit comes, just so you're not leaving seed behind. But it's a good way to get volunteers to come out if you tell them they can pick berries before they have to do some manual labor.
There you go. So when do the berries ripen then?
Our volunteer event was the beginning of August. That was maybe a week later than we would have liked it to be. So I would say end of July, early August.
Great time to get out and, start picking some berries and taking out bramble. Thank you, Will. So if anyone else has any questions, if you want to just pop them in the chat box. Next up, we'll have Amanda Barber. She'll present on her work in Cortland County. Amanda, you should be a co-host. You should be able to pop it up. Perfect.
Everybody see that okay then?
Yeah, looks great. Thanks, Amanda. Take it away.
So thanks for the opportunity. So my name's Amanda Barber. I'm the district manager here for Cortland Soil and Water. And this was a project that was not mine alone, but it certainly took the entire staff as well as a number of volunteers to get this done. We looked at opportunities to expand our invasive species management program. And if you don't know where Cortland County is, it's right in central New York. We have the tip of Skinny Atlas Lake in the county. We have a number of smaller kettle lakes in the northern portion of the county. Two major streams, the Sealick River and the Tyopneoga River, were at the headwaters of the Chesapeake Bay. And we have, some major transportation corridors, mainly being Route 81 that bisects our county, but also Route 41 and Route 13 that run through our county. So we feel like we're kind of at risk from an invasive species spread perspective. And our efforts have, other than working with the lakes to do invasive species management, aquatic invasive species management, our focus had been on aquatic invasive species spread prevention with some limited invasive species education and the use of IMAP invasives that we really wanted to enhance. So when we approached our project, it was sort of trying to start to fill some of the gaps that we had in what we would like to eventually develop into an overall invasive species management program. It's been, I think 2014 was the last time we had done any invasives mapping on our lakes. So we wanted to get back out to some of the lakes. We also recently had started to focus more on really our only county-owned park, Dwyer Park. It's about a 55-acre park right on Little York Lake, one of our lakes that I had mentioned. And we wanted to start to assess what was happening there and had applied to get some funding to do a forest management plan for the park. And we thought that this would be a great opportunity for us to take a look at invasives within the park. We also had been informed by one of our lake associations that they had identified some new invasives of concern from the PRISM's perspective. They had yellow floating heart. We had also recently identified and been attempting to develop some treatment strategies for starry stonewort. And we had since 2016 have done the aquatic invasive species spread prevention program, which included having boat stewards at both our public access points on the two lakes, Tildy Lake and Little York Lake, supporting the Skinny Atlas Lake program, as well as having stewards periodically present signage and resources available at other public launch sites on our rivers. Throughout the county, there's about another 7, I think, access points on rivers where we have a fairly robust kayaking and canoeing presence. So that program has been going, like I said, ongoing since 2016, and we had then applied for and received funding to install a boat cleaning station at Dwyer Park on Little York Lake. And we wanted to make sure that we were doing everything we could to promote that resource. And so we wanted to bring that in to this project as well. And then in general, we recognized that while we had done a fair amount of outreach to the lake property owners and to some degree those who utilize the lake as they come in and interact with boat stewards, but we have a lot more groups that we were beginning to realize that interacted with our water resources but may not be boaters. That people that were fishing, just people that were hiking near our waterways and water bodies, and then the general population. So sort of expanding from just aquatic invasives with, property owners and boaters, expanding that audience to other aquatic resource users, and then the general population. So that was kind of the focus of this project. So really multifaceted and multi-pronged project, but trying to, again, fill in some of the gaps in our program. So one of the things that we did, as I mentioned, was the Dwyer Park assessment. It was really an exploratory survey of the 55-acre park to record what invasive species were present and to understand more about their population distribution. We thought that this information would be really helpful and beneficial as we develop the forest management plan, and assuming that that forest management plan would potentially identify some treatment strategies. The approach that we took, and I give credit on a future slide, but Hannah Whalen was an intern who was working for us, and she did a fair amount of research on how best to approach this. And we decided to do transect lines. They're about 150 feet apart. We decided that just crossing the transect line may not be an effective way of identifying species. So we used a 30-foot swath, like 15 feet on either side of her transect. If she could identify it and see it, she mapped it as she was going. And we found 14 invasives, 7 woody plants and 7 herbaceous. Some of them we knew existed, some of them we did not. They were put in IMAP invasives, and we do plan to go back out when we do the forest management plan. We did find out that we were funded through the community forestry grants to get some resources there to do the management plan, which will be very helpful and allow us to be much more thorough in the plan than what we might have been able to do on our own. And we will be verifying these species and their locations again when we do the management plan, hopefully this year. The next assessment survey was our aquatic invasive survey on the lakes. We do have 6 lakes that we were looking at. We decided it wouldn't be feasible to do obviously all of Skaneateles Lake, but we were going to focus on the portions of the lake within Cortland County. So we went out and utilized volunteers or boats and kayaks where we needed to on our own. Did a rake toss sampling on a 50-meter grid where it was possible. We did have a little trouble with one of the larger Skaneateles Lake in particular with that process, but it ended up working out well in the end, and we're able to provide each lake association or each community their own map of their lake with maps indicating what species were present where, and all of that data was also uploaded into IMAP invasives. The surprises that we had were that in particular, Skaneateles Lake didn't realize that they had starry stonewort. And we did find one specimen of brittle naiad in Song Lake. Again, those were confirmed, and we are going to be going out and looking more closely, in particular at Song Lake, to see if we can identify any additional locations. And Skinny Atlas Lake, we continue to work with to see if they want to develop some sort of a treatment strategy for them. They, both lake associations, sort of a side note of this process was really opening up doors to expanded communications with the lake associations, and really has increased our opportunities to work and collaborate with them on other projects. So going forward, we're hoping to address some purple loosestrife, particularly on Goodale Lake. It's we call it a lake, but it's quickly converting to more of a wetland. As I said, we're going to be looking at Song Lake more for brittle naiad. Little York Lake will be continuing to do and expand on some success they've had recently with some herbicide treatments, as well as the use of some lake bottom blankets and hand weeding. We will be working with Skinny Atlas Lake and doing more effort to identify how extensive the population of starry stonewort is. We're not sure if it's just localized at the south end of the lake or if it does extend throughout the lake. And then, of course, we're going to continue to work with our other lake associations to do ongoing assessments. We've developed some protocols based on the PRISM's sampling, and we would be helping them to develop strategies for their own individual lakes based on where we know the populations to be and what we know they have there now, so that we can keep this information and mapping up to date. Our control strategies had been focused on the idea that yellow floating heart was present in Little York Lake, and it turns out that it was not a yellow floating heart. It was actually a native plant, and they also thought that they had European frogbit, which we were able to confirm that they do not. That's the good news. We were able to still assist them with their treatment and management efforts. They had done an initial variable leaf milfoil treatment in 2019. They had planned to do a second treatment in 2020, and then we're also going to do a pilot treatment of starry stonewort in 2020. So we did assist them with that pilot treatment, and are evaluating the impact, and we'll continue to evaluate the impact of that going forward. We do know that we saw fairly good treatment and a systemic effect of the treatment on the variable leaf milfoil and all the milfoils actually in the lake from the 2019 treatment. So we're hoping to see the same from the treatments in 2020. And we are actually planning to move ahead with them to do some additional treatments this year. The starry stonewort was also successful. And it was quite evident in our mapping where they had done the treatment. The treatment was done prior to our mapping. They did the variable leaf milfoil treatment in June, and then the starry stonewort treatment was in July. So we're continuing to monitor those results. We're overlaying mapping of where the treatments have occurred with the mapping that we did. And we'll continue to do that with our volunteer assessment programs so that we can document, hopefully, the successes of those treatment. And then we also had, as I mentioned, secured funding to install a CD3 waterless boat cleaning station, and have utilized various outreach methods to promote the use of the station to prevent the spread of aquatic invasives. From a public awareness perspective, as everyone mentioned, it was a little challenging this last year, but we were still pretty successful in finding other methods of doing outreach. We did do a mailing to all of the Kettle Lake residents, and we had great support from the Portland Onondaga Federation of Kettle Lake Associations to do that. That mailing and newsletter included information about our programming, information on how to identify invasives and where to go for additional help. We did 4 presentations. We had planned to do more, but we were still able to do 4 presentations to our highway superintendents, focusing on more invasives that they would be concerned about, as well as to an FFA class and two Rotary clubs here in the local area. All of them were Zoom presentations, but very well received, and actually maybe even more successful than trying to do face-to-face presentations. We are continuing to support the lakes, as I said. Our water quality specialist is now a member of the Skaneateles Lake Ecology team, and she's going to be continuing to work with them on the Starry Stonewort effort. And during Invasive Species Awareness Week, we did target emails. We identified a number of different ways that we could reach out through emails to groups. We tried to divide them up into groups that would be interested in similar invasives. So we took the important invasives list that the Finger Lakes PRISM has, and we broke it down according to those that would be more aquatic in nature or more terrestrial in nature, and that kind of thing, and put together a list with links to fact sheets, and sent that out all together. We sent out over 1,800 emails, which did generate a fair amount of interest in our Facebook page. Also, we did this during Invasive Species Awareness Week. It's on the slide, but we reached out to all of our ag producers and rural landowners. So our ag producers, we did through our own soil and water ag producers email list, but also through our cooperative extension had sent out an email through their newsletter list. And rural landowners are actually the rural landowners that have purchased trees and shrubs through our district seedling sale program. We utilize that email list to send the invasive species awareness letter to. We sent one out to all of our county legislators, all of the county department heads. The Lake Association sent them out through their entire email distribution list. We sent it out to our cooperative extension, who forwarded it to all of their 4-H club distribution list and 4-H members. The Boy Scouts in particular, locally, we had a contact who's a member of the Boy Scouts, so they sent it out through, I'm not going to remember the council name, but it was to their entire council. The email went out. State DOT was very interested in this when we gave our presentation to the Highway Superintendents Association, and they said if there's anything they could do to help us, they would. So we sent the information to them, and they forwarded it out to all of their Adopt a Highway volunteer teams, so that they were aware of invasives that they might find in, obviously, highway right-of-ways and road corridors. And then we were working with the Finger Lakes Trail, and sent one to them, that same email to them, which they forwarded out to their email distribution list. And then on our Facebook page, we had 7 posts. We did posts throughout Invasive Species Awareness Week, but we also did posts throughout the year. We had 5 blog posts during Invasive Species Awareness Week on our website. And then we also created an entire separate web page, which I don't know if this link is going to actually work, or we'll see if it does. And so on our web page, we created just a little bit of information and listed links to some various important invasive species. So that's a look at that. Now if I can get back to my presentation, right?
Technology is always fun. You probably resume your slideshow.
Well, my last slide was really just to say thank you to the PRISM, and thank you to all of our volunteers who participated and helped make the project successful. And it's just got my contact information on there. So
were you able to successfully upload the variable leaf population into IMAP?
yes, everything. There was a bulk upload of all of the lake survey information. So they should have that. If they don't have that, it's in their hands now. Let's put it that way.
Yeah, sure. And I ask because USGS doesn't have it, only has it as Cuba Lake, like an observation in 1992. And then IMAP doesn't have it. It has it, I'm trying to figure out which lake this is that they're confirming.
Yeah, so we didn't do individual points because we had so many points. They didn't want it done that way. We sent them all of the coordinates and all of the data, and they were going to do a bulk upload on their end, sort of from the back end. So I'm assuming that is on someone's desk as a
Yes.
upcoming priority.
We worked with IMAP before, so we know that that's coming. So on IMAP, it looks like there's a population in Shenango in the Balsam Swamp State Forest. So how dense is that population? It's 10 acres, 10.3 acres?
Variable leaf?
Yeah.
It's throughout the lake. It has been.
Really? Okay. And that's the only lake that you found it? Are you confident that it's contained to that lake, or do you think that there are other lakes where?
I mean, I think there's a concern that it's other places, but I'd have to go back and, I really would rely on Kathy to give me more feedback on what she found when she was doing that. But one of the things we ran into was we had originally thought, and forgive me if I pronounce things wrong, but we thought we had Cara. And then we found out that it was actually starry stonewort, and it didn't realize how difficult it was going to be to distinguish between the two. So that's just something that's come up the last couple of years in a number of the lakes. So
yeah, I think starry stonewort and Dave Carr can speak to this a little bit more directly, but I think starry stonewort has been known to be present in Kofokla lakes for a number of years. Is that right, Dave?
That's my understanding. Yeah, we've worked with Little York Lake now. Don is going to, from the association, is going to do a presentation for us in a webinar series in June. I don't know how long they've been dealing with it, but it's been a while, I think.
It's only been a few years that they've had a major.
That's good. Yeah.
And you're the one that came down to help us positively identify which was which when we were out there.
Well, yeah, I mean, I'm no expert yet. We're all learning still, but honestly, the Cara and starry are easy to confuse, hard to tell apart. But the method I use is I pop the stem of it, and if it's stinky, if it smells musky, it's Cara. It's usually muskgrass or something. And then of course, if you find the bulbil, it's a starry.
And it's easy
that's a giveaway, right? And then you're home free. But that's not always the case, as I'm sure you're aware. And
I think that's the difference, is that the positive identification that it was starry stonewort, took a couple of years. I think we thought it was the.
And that has happened many places. My project is across the Great Lakes Basin, and I've heard that story from places in Canada and other places that it was identified in some places for decades incorrectly, and then they realized what it really was. So it's tricky.
And for that, your control mechanism, are you looking to control it through hand pulling, you said, or control of?
Well, we've got to see. I mean, the conversations with the Lake Association is that they're not expecting the same effect on the starry stonewort that they had on the variable leaf milfoil. So the fear is that it is going to be back, and that they are going to need to do some sort of continued treatment. The hope is that some of our natives are beginning to take over where the starry stonewort has been knocked back. And we are keeping our fingers crossed about that. We have seen some native pondweeds that seem to be increasing in population. And so we're hopeful that they're going to be able to outcompete if we can get a handle on that starry stonewort.
Everybody is everybody, it's a broad term, but anybody trying chemical treatments in the same boat. They're all trying to find that formula to really knock down starry. And from a chemistry standpoint, I don't know much about this, but you're talking about algicides versus herbicides, because remember, starry is a macroalgae. So there's a whole different thing going on there too, is trying to get the formulas right in the water column and so on and so forth. And the experts in chemical application can tell you more about that, but it's hopefully somebody's going to come up with something soon because everybody's looking at the problem. You know?
And you'll hear more about it from the Lake Association, but that is, you know, we're watching to see what happens this year. And that's one of the reasons why the ongoing assessments are going to be important in the lake and keeping track of what those populations are doing.
Yeah, so it's great you guys are taking on that. It's a big thing to handle, but it's so important. It's really good research. So we need to hear
maybe we can look to Cuca Lake in their dash to see if that has any effect. Yeah, great.
Absolutely.
Well, thank you, Amanda. Is there any other questions? Go ahead and put them in the chat box. Carrie Marshner is up. And if we have any additional questions, we'll tack it on to the end. So Carrie, you have the floor. Thank you.
Thank you very much. Can you guys see my show here?
Yep, your first slide is up.
All right. So thank you for having us through this. And I hope I breeze through this quickly so we're on time. So I work with the New York State Hemlock Initiative. We're based at Cornell University, and we're all about hemlock woolly adelgid. And unless somebody puts in the chat that they don't know what hemlock woolly adelgid is or what we do, I'm just going to assume that you guys have seen one of these before and go straight to the updates. So this is the life cycle of HWA. They lay their eggs for the overwintering generation in like May, June, and then they overwinter, and then there's a spring generation that those eggs are laid in March, early April. They hatch right about now, and then they are moving on to lay the next overwintering generation. And the reason that I'm reminding you guys about this is that we now have active crawlers, which is the only life stage of this pest that's easily moved around. So if you are out surveying at this time of year, because the weather's beautiful and they're really easy to see right now, which all of those things are great, try not to go from an infested stand to an uninfested stand without really taking a lint roller to your clothes, maybe wash your hands, maybe go home for the day and, you know, change clothes before you go back out, because this is the time of year that we can move them around by accident. So this is the current state of HWA in New York. We've been looking at this map collectively for many years, and you'll see that a lot of it looks pretty similar, but I want to draw your attention to this sudden explosion of points in the southern Adirondacks around Lake George. This was a big find late last summer. A camper was up from the city camping at a campsite on Lake George, and he said, "That's like hemlock woolly adelgid. I have that in my yard. I didn't think it was up here yet." And he put it in IMAP, and that was why this big infestation was found. I believe we're at about 250 acres so far for this infestation. DEC started treatment in the fall. They're at this point trying to slow the spread because the cat is probably out of the bag, as it were, in this location because the infestation is so large. The other thing I want to draw your attention to is these 2 points. Just a couple months ago, the first point in the Slelo prism was confirmed. And then some of our volunteers were up in the Syracuse area looking in this area between the last known infestation and the new one. And they found the closest point yet to Syracuse. So this is probably going to be a fruitful area to look in the next year. HWA in the Finger Lakes, this is a little bit complicated because I put the negative points on here, but there's a reason for that. You can see that we have solid infestations, and lots of data through most of the county. And in this area where it's still negative, we have a lot of negative points in the northern portion that are showing that, yes, in fact, it's not here yet. Some of these are quite recent. And this year we started poking around down in this area, verifying some negative points. And we're looking to see if these 2 points, which were pretty old, they're from like 2014 to 2015, if we can still find HWA. We're just trying to nail down the leading edge down here. So you can see us through most of the places that we have hemlocks now, except the northern ends of the lakes to some extent. In the last 2 years, there's been a lot of work done on Skinny Atlas Lake last year by Steve Kinney and our Central New York HWA Hunters team. And this year by Dana Hall and the newly formed Owasco Lake Hemlock Hunters team. They did 17 surveys and counting all around Owasco Lake, found a bunch of HWA, and they've already started treatments in that watershed. So that's a big win. And thank you very much to Dana Hall for putting that together. The other thing I wanted to point out was we also have been working with groups in the Southern Honeoye watershed, trying to put together a similar survey team down there. We did an initial training this year. It was a little late in season, so we're hoping that we can get volunteers together to do some surveys to delineate what's going on here and figure out what the best next steps are. Every year we track overwintering mortality for HWA. We're probably done with that since the weather is warm, and we had the lowest mortality we've ever seen this year for HWA over winter, especially in the Finger Lakes, 20% mortality. That's incredibly low. Often our mortality is in the 90% and it still continues to spread. So this is an important piece of information for all of us in Finger Lakes as we're looking around. We should see a lot more of this really, really nasty, intense infestations because we've had 3 mild winters in a row now for these populations to build without that really helpful cold winter diet. A lot of what our organization does is biological control. We work with 2 insects, and the first one I'm going to talk about is the newer one, which are the Leucopus genus species that out in the Pacific Northwest do a lot of predation on HWA. There are 2 species. I believe this is Argenicollis and this is Pinniperta. Their larvae are this big. They're super tiny, but they take an HWA infestation like this and they just go through and just blow everything up. They eat the eggs, and we've learned that while they're eating the eggs, they also dislodge the adult HWA, so they're causing mortality on the older generation as well. These guys are active in the spring, feeding. I showed you that 2-generation chart to show you what the life cycle of HWA is like. These guys are feeding on the eggs of both generations. So this is what we hope will be a really useful biological control species. In 2020, we did releases at 9 sites, and all of the ones that are in green were in the Finger Lakes PRISM. This was in part a result of the coronavirus pandemic. We were really limited in our travel options, and that was great for the Finger Lakes for our releases. So you did a lot of releasing in our area last year, and that's great. We also now have 6, maybe 7 sites where we've verified Laricobius sorry, I'll tell you about that in a second. Our next species is Laricobius. This is the beetle we've been working with for years. We also did some releasing, as you can see, last fall of Laricobius in the Finger Lakes. And what I was going to tell you is that we've verified Herbaceous establishment in six sites around the state, and most of them are in the Finger Lakes because they really like those warmer temperatures. But one of them is also Harriman State Park. And last year we found them at Wells, where we had just released the previous year. So that's really good news. One of our new areas of research is eDNA survey. eDNA survey for HWA in those leading edge areas to see if we can pick up HWA without actually stumbling across it in the field. We're hoping that this will be a much more efficient way of searching for HWA in places like Swelo and Adirondacks, where there's not a lot of HWA already. We're also using this to try to search for establishment of our biological control agents, which would be great because if you think back to the size of that little silverfly larvae, it's really hard to find those suckers in the field. So this work was started by Anish Kurtain, who worked with us for a year, and then he has moved on to his next thing. And just today, Feng Zhou has joined our team. So he's just getting up to speed, and we're collecting our spring samples to search for establishment at a handful of sites, mostly in the Finger Lakes. So hopefully we'll have some news for you on this next time I give an update. Our collecting season was a little odd last year because remember, all of our biological controls come from the Pacific Northwest. So with travel restrictions, that was pretty difficult. We had some really excellent collaborators at West send us material last year during the early coronavirus lockdown. And then this year, Mark just picked up and moved to the Pacific Northwest for 4 months. He's still out there collecting foliage for us and shipping us back so that we can do our biological research and releases. So hats off to Mark. Another thing that we've done a lot of work on that has moved forward a lot in the last year is our supportive management planning. Most landowners don't have enough funds to manage all of their hemlocks. And so they have this heartbreaking question of which trees do we save? Because generally chemical management is the only way to go at this time for keeping your hemlocks alive. We've worked with a lot of collaborators, including New York State Parks, New York DEC, and several other folks to put together this prioritization tool. And we finally have it published, and we're now doing trainings for this tool. So that's something that we can do for you, in addition to the general HWA talks. So if you have a group of landowners who are trying to figure out what to do about their HWA situation, we can offer this tool and walk them through it and help them think through what's the best way to spend their money. Another thing we're working on is updating the best management practices for HWA. One of the big questions that people have when they treat is, do I use just imidacloprid or do I use a tank mix? And we have some updated recommendations for that, which we'll be posting shortly. And with that, thank you. And thanks for the opportunity to talk.
Thank you so much. I'm super appreciative. We all are of the fact that we got a little extra bonus since COVID. We were able to get bio releases, and that's really great. I know that I was out with the Hemlock Initiative. We went to Webster Park up by Lake Ontario, and we were able to capture some populations of Herbaceous, and that was a great sign for the sustained population. And so that's always a good thing. And one of the 2021 funding opportunities will be to treat Hemlock Willia Delgid at Camp Cutler, which is a Boy Scout of America camp. And that's in the Honeoye Lake watershed. So we're looking forward to seeing hopefully good results there. So we have about 3 minutes left before the end of this meeting. Does anyone want to unmute and have any questions for the presenters, or any just comments in general? And my son is playing Xbox next door, so you could clearly hear that. Only told him 7 times not to. All right, well, not hearing anybody speak, we want to congratulate you all on great work that you've done for invasive species throughout the region. We will be looking forward to participation on the working groups or steering committee. Please reach out if you're interested in participating in those. And Mary has indicated that she had 3 calls for HWA in Livingston County just last week. So yeah, it's pretty bad there. So looking forward to collaborating with all of you on the call. Hopefully we can see you in person for our next meeting. Have a wonderful field season and reach out for anything invasive species related. We're here as a resource for you. So thank you. Have a great Monday.
Thank you.
Thanks, Hillary.
Thanks, Katie. Looking forward to working with you.